Good evening, everyone. Good evening. I'm Daniel Gordon, Professor of History and Associate Dean of the Commonwealth Honors College. It's my privilege again to introduce the faculty lecture for students in Honors 291A, Ideas That Change the World. Professor Sheila Patek received her bachelor's degree in biology at Harvard and her PhD in biology from Duke. She's currently Associate Professor of Biology here at UMass Amherst. Professor Patek has a very long and impressive list of publications with articles in the Journal of Experimental Biology, Integrative and Comparative Biology, Evolution, and many other important journals. She's received numerous honors, including the Brilliant 10 Award from Popular Science magazine. Her research has been funded by the National Science Foundation, the National Geographic Society, the Beckman Institute for Advanced Science and Technology, and many other organizations. Professor Patek directs a team of researchers in a lab called the Patek Lab. The lab has a beautiful website. I encourage you to check it out on your own. It includes a set of guiding principles, and I found reading these inspiring. I also noticed that they overlap with the goals of Rachel Carson, whom we study in Ideas That Change the World. So I'd like to read 2 of the 5 principles of the Patek Lab. Principle number 3, probe questions and problems that transcend traditional fields of science and stimulate integration of scientific approaches and perspectives. Principle 5, make our findings accessible to the broader public, such that we stimulate interest in science, increase knowledge of biology, and thereby inspire a commitment to and protection of the biological world. Clearly, with Professor Patek, we're dealing not only with a great mind, but someone who's preoccupied with the ethical and public issues of the type we've encountered in Silent Spring. Please join me in welcoming Professor Sheila Patek, who will speak about the unsilent sea.
Thank you so much for the really nice introduction. I hadn't returned to my guiding principles for a while. It was good to hear them and be reminded of what we're working toward. Thank you so much for coming out tonight, and I wonder if I should turn off this one. There we go. And especially on such a cold and somewhat miserable evening, and it's really an honor to get to talk to so many UMass undergraduates and other members of the UMass and Amherst community. When Priscilla Clarkson invited me to talk here tonight, she mentioned that she'd thought of me because of the reading that many of you are doing for your Commonwealth Honors College seminar of Silent Spring. And this inspired me actually to go back and read Silent Spring again. I hadn't read it for a few years. And not only that, but actually read another one of Rachel Carson's book, actually the first book that made her famous, which is called The Sea Around Us, and that's where that quote is from. And not only did it inspire me to go back and read this, but it inspired me to try to create a lecture that pulled in some of the themes from Silent Spring. And I entitled the lecture Unsilent Sea as a reflection on her title, but also with the notion of trying to pull in some of her broader principles. And so what I'll talk today about is this fundamental love and fascination we have with the natural world, which Rachel Carson expressed so beautifully in her book. I'll talk about the specifics of some of my research, which may seem not connected, but I'll try to connect it for you, about the physics of the environment in the ocean and sound, the consequences for sound production in the ocean. And finally, again, trying to emulate what Rachel Carson did so well in her book that really did change the world and still is very relevant today, to try to inspire you to think about the ocean and the sound in the ocean, and perhaps join in with my wish and goal to make sure it doesn't become a silent sea, just as Rachel Carson was talking about when she was hoping that we wouldn't have Silent Spring, which some people were having, no dawn chorus in the 1960s because of the over-application of insecticide. So it's not going to be a depressing talk, actually. I've actually made people cry before while I talk, and that is not the goal today. I'm actually going to try to inspire you to be into this stuff. So I'm going to start by painting a picture for you of this transition, this difference between life in air versus life in water. And this is a picture of one of the places where I do my research. It's Catalina Island, right off the coast of Los Angeles. And if you were going to be sitting right on the edge there out, you know, looking out over that beautiful bay, you would hear maybe a bird singing or a couple of crickets cheeping, but it would be pretty quiet. If, however, you went down to the water and you stuck your head in the water, and by the way, for those of you who are not from California, that water is very cold in the time periods when we're there. You stick your head in the water and you would hear a cacophony of sound. And it goes on all the time, day and night. Why is it so loud in the sea? I'll play you a clip in a minute of what it sounds like. It's because of the physics of water. So sound in air attenuates, it fades away very quickly. It doesn't travel very far. We don't feel vibrations very well. But in water, sound travels 5 times more quickly than in air. The range where you can feel vibrations is 5 times larger. Sound fades away very slowly. And that means that, just as a way of analogy, when you walked here tonight and you passed someone on the sidewalk, I'm sure you didn't physically feel them unless you bumped into them. However, if you were out snorkeling and you passed someone, you would feel them with your body go by. This means that when we listen to the sea, we're hearing everything. We're hearing organisms moving, we're hearing animals chewing, we're hearing animals communicating. It's a really noisy place. And one of the fun things that I found in my meanderings through Rachel Carson's writing, she wrote this quotation in her The Sea Around Us book, and she said, one of the most extraordinarily widespread sounds of the undersea is the crackling, sizzling sound, like dry twigs burning or fat frying, heard near beds of the snapping shrimp. And indeed, the sound I'm going to play you is dominated by this little critter, the snapping shrimp. And I'll play you. This is a recording I literally made in the water that you're looking at. And it goes on all the time. And as, of course, you'd expect with someone with the writing skills of Rachel Carson, that is actually what it sounds like. The crackling, sizzling sound. What she did, though, with this quote, of course, not intentionally, is she set up this lecture incredibly well. So let me turn this off. I could have probably been a little bit more elegant with not having to turn around while I'm doing this. But she next wrote, "The shrimp are forever clicking the 2 joints of this claw together." And this is great, because for 50 years, that's what people thought that they were doing because they didn't understand or didn't think about the fundamental differences between moving quickly in air versus water. This is not correct. It's not her fault at all. But there's this big claw on the snapping shrimp, and if we take a look at it from the side view, there's a little one part that's gonna, we're gonna slow it down. This was filmed at 40,000 frames per second. It was published in Science in the year 2000. And these folks decided they were gonna film these animals making this movement just for kicks and to figure out a few things. And what they saw immediately was that, come back, as the jaw is closing, this is slowed down tremendously. It closes, but it shoots a jet of air. This animal's completely underwater, and that was an air bubble that just came out of the claws closing. Look at it again. So it closes, and out shoots a bubble of air. This is called a cavitation bubble, and when that cavitation bubble collapses, it emits an enormous burst of sound. So in fact, it turns out that the snapping shrimp are not clacking their jaws together, making these sounds over and around the planet 24 hours a day, 7 days a week to make that sound. They're actually shooting incredibly and energetically intense vapor bubbles at each other, at prey. It's their whole mechanism for catching prey and for communicating with each other. The reason why I bring this up, not only because it's cool, we're going to talk a little bit later about why cavitation happens, but it points out the hallmark of the research that I do, which is to say, well, we have this great terrestrial view as scientists, but let's see what happens in the water. Now, what I'm going to do today is give you 3 vignettes about sound in the sea. I'm going to start by talking about a more traditional sound in the sea source, which is in lobsters that produce sound to scare away predators. Now, when I say traditional, anything I do is non-traditional, so this is more traditional on my scale of traditional. So, for example, traditional sound in the sea would be whales or dolphins. Of course, they constitute probably 0.05% of the sound in the sea, maybe not even that much. The rest of it is critters like this. We'll look at cavitation bubble formation, the effect of the relationship between movement and sound in the sea, and mantis shrimp. And we'll finish by talking about a really ambiguous sound that's also produced by mantis shrimp at the end of the talk. And consider, again, the sound in the sea not just as an interesting area of physics and biology, but also as a really major frontier. So let's take a look at stick-slip sound production in spiny lobsters. This is a picture of the California spiny lobster, and since all of you, or many of you, are from this part of the country in the Northeast, you're more familiar with the lobsters that have big claws on them. And by the way, those lobsters, those Maine lobsters, produce sound. They have a sound-producing muscle in their head, and this has been known for a very long time. These sound-producing lobsters do not have claws. They're spiny lobsters, and they're the kind where you eat the tail at red lobster. And so they don't have claws, but what they do have are really long spiny antennae, and at the base of the antennae is a sound-producing structure. And when they get really annoyed, they make a lot of sound. I'm going to play you a sound that was recorded by an undergraduate in my lab, and all she was doing was holding this lobster. That is the sound of a mad spiny lobster saying, "Let me go." Which I don't know what exactly she was doing, but that's one of my best recordings of a mad lobster. And what they do is they produce these sounds when they're trying to scare away predators or trying to get an organism to let go of them. So that was one lobster, but imagine a den of lobsters, which is how these animals usually live, and the cacophony of sound that they produce when they're feeling threatened by a predator. So what is this particular kind of sound production? So we call it a rasp, and these rasps are used for scaring away predators. It turns out that you can name a sound, which we've done in my lab on several occasions, but it's really hard to pinpoint exactly how it's used, even something that seems as straightforward as a sound that's produced when an animal is annoyed with you or trying to deal with a predator that's attacking it. So one possibility is that the sound production is to scare away a predator. So an unexpected noise and you go, yikes, and that gives the animal a chance to get away. Another possibility is that these sounds are reminding potential predators that, oof, I had an experience with these awful tasting creatures. It made sound, and now when I hear the sound, I'm not going to go eat it. So kind of an aversion. So one of the things that we have to do when we go and we look at sound production in the ocean, where people haven't looked at the behavior and we don't know the function, and we actually have to go out and do experiments to figure out what the sounds are for. And what we found actually is that it's not a yuck sound, it's more of a yikes sound. So they make the sound when they're actually physically being held, but not in anticipation of being held. So obviously if you're going to say, "I'm taste terrible," you're going to tell your potential predator that before they taste you. So they're not doing that. So that's a rather colloquial way of talking about a rather complex set of behavioral experiments. So we go out to Catalina, and I have always brought undergraduates with me, and we've done a number of experiments, both on land in tanks and out in the ocean. One of the things about these lobsters is that they're nocturnal. So we get on our wetsuits, hoods, gloves, the whole 9 yards, jump in this water in the deep dark of night with the seals and the sharks, and we interact with those lobsters and record their sounds and look at what the nocturnal world of sound is like for these animals. Now, another part of working on a sound-producing system, which not much work has been done, is to figure out how they do it. Spiny lobsters, as I mentioned, they have this little piece at the base of their antenna, and they rub it over a little file on their head, and they move it back and forth, and it produces sound. This has been known, actually, the first, as far as I know, documentation of sound production in lobsters was by Athenaeus in 300 AD. And this is because these are audible sounds, and fishermen, when they pick these animals up, or they're squeaking away. So people have known that, but it wasn't until I started working on it that we did some real biomechanical tests to figure out why and how they're making these sounds. And we're just going to do the tiniest bit of morphology just because it's just so cool. This is what the file looks like. It's about 2 centimeters long, totally smooth surface when you look at it, just not with a microscope. And then there's this funny little chunk of soft tissue that rubs over it. And so it rubs in that direction. We can take a close-up with some microscopy, and these are these ridges that are on the soft tissue, and they rub over the surface of the file. And this is at the same scale. Now, I hope you're finding this extremely non-intuitive, but hopefully not boring. But this is really weird. There are these sort of fingery soft tissue projections rubbing over teeny, teeny, tiny shingles on a surface. We can blow it up. That's what it looks like, really close up. But there wasn't any way to understand this in the biological bioacoustics literature. The way we understand the production of those raspy sounds is from crickets and other systems like the washboard, where you have a hard pick that runs over a series of bumps, and every time that pick hits a bump, it generates a little burst of sound. So that's our traditional way of understanding sound production in arthropods, but our lobsters don't give us that. It's got smooth and smooth. How do we solve this problem? Well, oftentimes what I do, and less so now that things are so digital, but I just hoof it on over to the library and start looking through books. And the musical acoustics literature, which is really fascinating, hopefully there are a few musicians in this audience, had a great hint. And the hint was with the way bowed stringed instruments work. So as you guys probably know, the bow and the string on a violin are smooth macroscopically. Yet somehow, if you slow down this whole system with a high-speed video, there is this sticking and slipping motion of the bow over the string on the violin, so it holds on and release, hold on, release, hold on, release. This is called stick and slip friction that allows this to happen. And so my thought was, well, maybe this is what the lobsters are doing. Maybe they're actually doing some kind of frictional interaction to allow them to produce this sound. Well, a whole bunch of experiments later, and I was able to show that indeed they use the same underlying frictional mechanisms that violins use to produce sound. And this was the first discovery of an analog in biology to the underlying mechanism of sound production in bowed stringed instruments. Just sitting there right in front of us. How many billion spiny lobsters have been eaten before anyone actually thought deeply about their sound producing mechanism? I can't stand to think of it, but there it is, really interesting finding. The other thing that's cool is if we place this in the biology of these animals. So they have to molt, they get very soft during the molting period, and because they don't use a hard mechanism, like a cricket or washboard mechanism, they're able to make the sound at any time during their life cycle, even if their bodies are totally soft right after they've molted, which of course is pretty handy if you're using the sound to scare away predators. The other kind of cool thing is it turns out that stick and slip friction is something we're pretty familiar with in daily life. It happens to be stick and slip models are used for understanding earthquake, the interaction between earthquake plates. This is the sound of squeaky chalk. So if you get the right pressure and velocity on the board, that's stick and slip. It's the same sound as your finger rubbing on a balloon. And one of the things that I've continued to come back to is, well, first of all, why don't other biological systems use this? But how have the lobsters evolved to actually control and produce these sounds through evolutionary time? So we've looked a lot at the diversity of lobsters, and I just love this picture because it highlights just the beauty of these animals, which isn't often talked about, such a major food source for the planet. This is the face of a lobster. I took this picture at a fish market in Japan. The little files are blue and the plectrums are pink. Those are the eyeballs. This is a deep sea lobster. I took this picture at an aquarium in Japan. It also has a sound producing apparatus up there at the head, and another beautiful one there. They're just spectacular animals, each one with a slightly different mechanism of sound production. So to understand this, I built an evolutionary tree. I won't go into the details about this, but actually looked at the evolution of the sounds, how it varies across the family, and how they generate these different sounds with different morphology. And just for kicks, I'll play you two sounds. This first one is a sound of a spiny lobster recorded in Japan. And this is one that was sent to me by a fisherman in Ireland, sent me the lobsters that made this sound. And you can hear the differences. There are all these different sounds. Why do they have so many different sounds? Well, one answer, of course, is that these are ancient creatures. The source of life, of course, is from the sea. Not surprising. These animals have been around. The first fossil evidence of spiny lobsters is from 220 million years ago. This is approximately what we think that the Earth looked like at that time. The amount of time is amazing, and this brings me to my first quote from Silent Spring. And one thing Rachel Carson really understood deeply about biology is time. She wrote, "Given time, time not in years but in millennia, life adjusts and a balance has been reached, for time is the essential ingredient, but in the modern world there is no time." 220 million years to produce 45 different sound-producing mechanisms that they use for deterring predators. And our time on this planet is barely a blip here, and yet our impacts on spiny lobsters around the world are massive. I mean, just in that marine lab that I was showing you a picture of, that's a marine reserve. There are an abundance of spiny lobsters in there. If I go outside of the boundaries of that marine reserve, even 20, 30 feet, I can't catch a lobster after lobster season is opened. I mean, it takes me weeks of trapping to get maybe 20 lobsters. So our impacts on these animals are incredible, and we're just barely scratching the surface of our understanding of their biology. Many countries around the world that don't have management of the lobsters, the best you can get are like 3-inch sized lobsters, where in places where people have been talking about historically finding lobsters that were 3 feet long. So our pressures on the system are tremendous, and such a drop in the bucket relative to the time that these animals have been on the planet. Something Rachel Carson deeply understood when she saw the impacts that we were having in the 1960s. And another really cool thing is that there's evidence of this history. So there are fossils of spiny lobsters with the actual sound-producing apparatus preserved. And one of my dreams is to do something that some of my colleagues did a while ago with bush crickets. So they understood the morphology of sound production. They had fossil information, and they could actually figure out the sounds of ancient bush crickets from the fossil information. One of my dreams is to figure out what the oceans sounded like maybe 45 million years ago by looking at this connection between morphology and sound. So now let's shift gears and not talk about sound that's intentionally, if you want to use the word, used for communication, but sound in the sea that's a product or a consequence of movement. And we'll look at this in cavitating hammers and mantis shrimp. So Rachel Carson set up, again, I got so excited. It was fun going back and reading this book. I think it must be my 3rd or 4th time reading this book, this time with a different idea in mind. And when I got to this passage, I was like, oh, perfect. So she writes, "We are seldom aware of the protection afforded by natural enemies until it fails. But we see with understanding eye only if we have walked in the garden at night and here and there with a flashlight have glimpsed the mantis stealthily creeping upon her prey. Then we sense something of the drama of the hunter and the hunted. Then we begin to feel something of that relentlessly pressing force by which nature controls her own." She is talking about the praying mantis in her garden. A praying mantis has these wonderful predatory appendages, and it stays totally still until a prey item goes by and it wings out and grabs it and eats it. And she captures in that this like ferociousness of that little green insect and the excitement of seeing it and this dynamic predator-prey interaction that characterizes the ecology in biology. Our mantis shrimp, the organism that I spend a heck of a lot of my life studying, are named after praying mantises because they also have a powerful predatory appendage that they use to capture prey. And we're going to talk a little bit about this. And of course, being a mantis shrimp obsessed individual, I think they're even cooler than praying mantises. So let's take a look at them. So this is I'm going to show you a couple videos of these animals in action. This is an animal called the peacock mantis shrimp. We find them in Indonesia and Australia, and are about the size of a cigar. So they're pretty good sized animals, and they zip around and they love snails. So this is an animal processing a snail, and it's going to hammer it with its appendage, and it's going to break it open. So they smash it, and the snail shell broke. Hopefully you saw that, and then I'll show it you to you again in a second. But what's missing from this image is the sound. So let's look at this again, but now I'm just going to basically turn the sound on. This is just a standard video camera outside of a noisy tank in a noisy room. No special effects, and now let's listen to what they do. Boom, right? You think, wow, that is the hammer hitting the snail. As it turns out, that's only a small fraction of the actual sound that's being produced. Most of the sound that's being produced during that movement is, again, that infamous cavitation bubble. And so we discovered, by the way, and I should have mentioned this, to open one of those snail shells, I could jump on one all day and probably not break it. So we have to, as humans, would have to use a hammer, a good hammer blow to break it open. And these animals are often able to crack them with 1 or 2 hits of the appendage. So I decided I wanted to film them and see what they're doing. And to do that, you have to use some serious high-speed video. And as we filmed at 5,000 frames per second, there's a snail shell wired to a stick here, which helps us set up the shots. And there's the hammer of the appendage that's going to swing around and hit it. And when we first filmed this, by the way, and if you look right here, you'll see what made us tremendously excited. It's the formation of a cavitation bubble at the surface. So you sort of see a flash of light. I analyzed all these videos, which I won't go into the details about, but to give you a sense of the speed of this movement, they do this movement in under 2 milliseconds. So that's, 150 of them would fit within a single eye blink. Of course, they don't do it that, at that rate, but it gives you a sense of the shortness of the duration. This is out of the range of eye blinks. They can get up to 24 meters per second, which is about 45 miles an hour. And this means that they have an acceleration of a bullet. Our lowly shrimp accelerating with a bullet. It's no wonder they can generate these forces to break snail shells that we need hammers to do. But because they are moving in water, they can cavitate. So let's take a look at an even slower paced video. And there you can actually see that air bubble spreading over the surface of the snail. And then we can take an even closer look. So this is called cavitation. Cavitation has been known for a long time because if you move something through the water very, very quickly, it generates this thing called the Bernoulli effect. It generates an area of low pressure next to an area of high pressure because of the different speeds of the water. And at that interface, the water molecules are pulling away from each other, and that causes a vapor to form. It's kind of like if you put a pot on a stove of water and it starts to boil, right? It's generating vapor bubbles because of the heat. Here it's generating vapor bubbles because of low pressure. But because it's surrounded by the pressure of the ocean and the pressure of the water, the bubble can collapse. And when it collapses, it emits heat equivalent to the surface of the sun, 6,000 to 7,000 degrees Celsius. It emits light, and it emits a huge amount of sound and pressure. And I'm not making this up, by the way. There was a paper a couple of years ago where a group had actually thought that they had observed nuclear fusion at the instant of cavitation. Eventually it was debunked, as is everything about nuclear fusion, but it gives you a sense of the intense energy of this bubble collapse. It destroys boat propellers. These are streams of cavitation bubbles coming off of a propeller. And of course, the mantis shrimp itself experiences damage from cavitation, but they have the advantage of being able to grow a new appendage. However, in the meantime, they can strike tens of thousands of times without catastrophic failure. And if this was a scientific audience, or if you're welcome to talk to me about this afterward, there was a huge paper that just came out about how they're able to resist these intense impacts of their appendages through the design of the actual appendage itself. So what I wanted to know when I saw this, I said, well, that's kind of interesting. How much force can they actually produce? And does the cavitation bubble help them break open their prey? So we would present them with a force sensor, and we can record the sound of cavitation with a hydrophone, an underwater microphone, because cavitation has its own acoustic signature. And they're aggressive animals, and they'll come up and give it a good whack, which this one will in a moment. and then we get the force data. And I can show you a slightly different view here. This is the force sensor, and you'll see the hammer come around, slowing it down even more, and the cavitation bubble forms, and here's a single image of it. So there's that cavitation bubble right there. Wow. However, it happens at extremely short time scales. When I did this, my colleagues were so skeptical that I had to go, I felt like around the planet 3 times to convince everybody that this was real. But it is. Filmed at 100,000 frames per second. Your home video films it at maybe 60 frames per second. This allows us to look at very short time scales. So the appendage is hitting the sensor right now, the cavitation bubbles form, and then we can actually watch them collapse. So ultimately, I was able to link up these videos with the force sensor. So what I'm going to ask you to do is just look at the video up here, again filmed at 100,000 frames per second, and watch the force trace that emerges. And again, what I'm trying to do is show that there's an association between cavitation bubble collapse and force production here, which had not been shown before. So there's impact, and right at the moment that the cavitation bubble collapses, there's a peak of force. When I show my data, I try to do something that's sort of a typical strike. This is fairly typical. Cavitation bubble is producing half the impact of the hammer impact itself, but the hammer impact itself is massive. So these animals are producing thousands of times their body weight in peak impact force. As far as I know, this still holds the record as the highest force generation of any organism that's been measured on the planet. So when we say that on average, half again of that force is being produced by a cavitation bubble collapse, that's impressive. And many of our traces, the cavitation bubble actually exceeds the initial impact. So let's put this in context. Mantis shrimp have 2 appendages. When they strike, they get an impact, and then they have a cavitation bubble and impact and a cavitation bubble. A hugely energetic event. And we can compare this to a shark that eats snails. The horned shark actually goes around and picks up snails and chews on it. If you look at the scale bars here, 200 newtons, and by the way, I chose a pathetic mantis shrimp strike to make this comparison so the shark biologist wouldn't feel too bad. And this is the highest peak force that a horned shark produces during in this study that these folks study this in. If you look at the time scales, the entire mantis shrimp strike happens right there. Note that the peaks are about the same, but let's compare the mass of these animals. This is a 3-kilogram horned shark. Its very best is approaching 200 newtons. A 40-gram mantis shrimp is exceeding many times that, 200 newtons, to generate an extremely potent force. And all of this is because of the movement happening in water that permits the production of cavitation and the amazing building blocks of the appendage itself that they're able to store up and release massive amounts of energy. When we made these discoveries, we basically were able to move the field, which has been over here for history, everybody thinking about chewing and breaking movements like that through crab claws or jaws, and say, wow, let's look at this. This is a totally different way for predators to behave in the ocean. And they can use the power of the water in which they're located to produce unimaginable amounts of forces. And so it's been really fun since we published this paper to watch many different groups of engineers in particular, but biologists as well, trying to start to disentangle this discovery of these incredible forces produced by the animal, the mechanisms by which they do it, and the use of the power of the fluid medium in which they live. So, very different perspective. Just again, from Rachel Carson's perspective, the first fossil evidence of mantis shrimp was 300 million years ago. That's what the planet is thought to have looked like. Hammers showed up around 50 million years ago. The sort of original mantis shrimp, many of them looked like this. Instead of hammers, they captured their prey, and they still do. They're the most speciose kinds of mantis shrimp, and by the way, we have beautiful examples of it here off the coast of Massachusetts. So they're called Squilla impusa, although this is not a film of them. And what they do is they hide in a burrow and catch stuff going overhead. So this is a slow motion video of a spearing mantis shrimp just catching a piece of food that's overhead, and they reach up and grab it. So a different strategy with the same building blocks. And again, think about the time here. 300 million years of evolution to generate some of the most extraordinary materials and movements and forces that we know of in the world. So finally, I want to end on a sort of discovery note. and the ambiguities and the challenges of looking at sound in the sea as a frontier. And this is the stomatopod rumble. So this is another mantis shrimp, and this guy's about 1.5 foot long. And let me tell you, when they strike, you get out of the way. These are the only ones that actually scare me. All the other mantis shrimp I'm okay with. They live about 60 feet deep in California, in the southern part of California, and they have this wonderful pink spot on the edge of their body. It turns out mantis shrimp can see way better than we can. They have some of the best vision of any organism on the planet, and they can see polarized light. Turns out this little pink spot is polarized. Now, if we have a polarizing filter, if we move the filter around, that spot flashes pink and white for us. But they can actually see the angle of the light coming off that pink spot. Now, what's really cool is that turns out they also vibrate that pink spot. And let me play you what it sounds like. This is going to be scary, so if you're falling asleep, now's the time to wake up. And I'm sorry if it's really loud. Sometimes it comes out really loud. That is really the sound that these animals are making. That is not a joke. And I will say that when I had sort of been on a quest to record that sound, and I'd gone all around, different field sites to try to, there had been anecdotal reports of it from fishermen. And one day I was having a really bad day. Some of you might work in labs here at UMass. And I was just like, darn it, when am I ever going to get data on this project? So I got my headphones on my recorder and my hydrophone. I said, darn it, I'm just going to walk through the whole lab and stick my hydrophone in every single tank with a mantis shrimp, and I'm just going to do it. And that was exactly the recording that I got when I happened to just stick it in the tank with one big mantis shrimp. And it was like intense. It was a memorable moment where I was like, this is the first time someone has recorded this animal making sound. 300 million years of evolution, and there is no documented recording of these animals producing sound. And here it is. So we named it the rumble, after the sounds made by elephants and whales. It's the wrong frequency for a little animal. It doesn't make sense to have a small animal producing such a low frequency sound. One possibility is it isn't actually a sound, that they're actually doing some kind of visual communication. So we went back to the field and said, what are they doing down there on the ocean floor? This is a picture of a mantis shrimp in its burrow in the real world, and this is a male, and he is very proud of his den. He's actually decorated it with some shells to attract the girls. So he's doing a good job. And it turns out, so we put hydrophones down there. We actually have some recorders that we can leave down there for weeks on end. And this is what we heard. You have to listen carefully for this one. I don't know if you could hear that. Wow. Again, a memorable moment when we pulled these recordings out and realized that we had recorded these animals basically, as far as we can tell, somehow talking to each other or calling back and forth. And this is again an exciting moment because we have almost no understanding of how animals that are living in the ocean floor are communicating with each other with sound. We don't have the answer to this yet, but I will say this brings me to the final message that relates to Rachel Carson's Silent Spring, which is what happens when a boat drives by. And I'm not talking about a big boat here, like a ferry. I'm talking about just a little motorboat that went by when we were recording these animals. The sounds that we are putting in the ocean are incredibly intense. The person who I worked with on this project, Erica Statterman, she was out recording with me one day and she was diving, and when a cruise ship came into the harbor. And she said she was down there and she was working with these animals, and her whole body was vibrating so intensely. She's like one of the most calm divers I know. And she said it was impossible to think, it was impossible to stay calm because her whole body was just stuck in the vibration of the motors of that boat. And she actually was so influenced by that she's now gone on to graduate school and is studying the effects of anthropogenic noise on organisms. especially coral reef larvae, which orient to sound. And if you obliterate the ocean with sound, it destroys the acoustic habitat. So we're working on this now, and I partly put up this picture of Boston with the ferry because the Massachusetts should be pretty proud of itself, because I think it's one of the most progressive ocean acoustic places I know of. So some of you may be familiar with Stellwagen Bank, which is an area where there are a lot of whales. And it was recognized that the amount of noise that the boats were making coming out of Boston Harbor was obliterating the ability of the whales to communicate with each other. So what people have done, and it's active today, is they're constantly monitoring where the whales are. And they actually reroute boat traffic so that the whales are able to continue to communicate and to have healthy populations, because it's so fundamental to their ability to survive. So of course, this is a different aspect that we're working on in the lab now, and looking at that. And in fact, the person who did all this Stellwagen Bank stuff emailed me today. I was totally, I work with him on the mantis shrimp stuff, and he wrote me an email about all this stuff, and he wrote PS at the bottom, and he said, I think that this is extremely important. We have no idea what is happening with animals in the actual substrate of the ocean. And this is the project that he's chosen to work on with me to try to solve this and figure it out. So Rachel Carson wrote, to the problem of sharing our Earth with other creatures, there runs a constant theme. The awareness that we are dealing with life, with living populations and all their pressures and counter pressures, their surges and recessions. So she's saying we gotta understand the whole thing before we mess with it. So I wanna talk just for about 1 more minute about the kind of biology that I've just talked about. And this biology is called organismal biology. We're looking at whole organisms. Many of you are inculcated with the notion that everything valuable about biology has to be at the molecular and cellular level. But I want to tell you why studying whole organisms is important for today. It matters because we can learn things from 200 plus years of engineering that's already been done through evolution. Our own faculty here at UMass have made major discoveries this year. This is Al Crosby in polymer sciences and Duncan Urshik in biology, and Mike Bartlett, who's a graduate student in polymer sciences, who led this work. And they made a major discovery of mimicking the fantastic adhesion of geckos for manufactured products that are much better than anything we have on the market. And we're working in my lab, led primarily by Suzanne Cox, to figure out whether there are ways that we can learn about how mantis shrimp control cavitation, and whether we can use this to inform our own design of fast-moving objects. I think it always needs to be reminded that our best model medical systems in biology today, the fruit fly, zebrafish, and mouse, the only reason why they help us solve medical problems is because there were biologists who studied the whole organism, its behavior, made key observations, and then from that we could build two systems that save lives every day. We need to understand our place on the planet. This is a phylogeny, an evolutionary tree. We are nothing, that's us right there. This is the blue planet. Most of our planet is ocean, and we know stunningly little about what's in there. And then, taking the words from Jacques Cousteau, we protect what we love. When Rachel Carson wrote, who has decided, who has the right to decide that the supreme value is a world without insects, even though it be also a sterile world, ungraced by the curving wind of a bird in flight, the decision made during a moment of inattention by millions to whom beauty and the ordered world of nature still have a meaning that is deep and imperative. She is saying, don't just sit there and watch this happen and let other people make decisions about things that you care about. You've got to do something. And she is, when I saw that you guys had chosen this for your book, what an amazing decision. Rachel Carson was just somebody like you and like me who wrote a book that changed the world. Because of her love of the biological world, she could articulate this and galvanize people to action. If I dare say it, I think our present day Rachel Carson is Sylvia Earle. She wrote a book, The World is Blue, and others. She is an incredible scientist, and she's been termed her deepness by many people, and she is the voice for conserving and preserving the ocean. So if you're at all inspired by this lecture, read this book. It'll really change the way you view our need for a healthy ocean. So, Unsilent Sea. Hopefully some different perspectives on Silent Spring and Silent Sea, what it means to produce communication sounds like a lobster that's like a violin, the first observation we have of an analog to a violin, the incredible impacts and sounds produced because of fast movements in the ocean, and then the wonderful ambiguities of a newly discovered acoustic system. What is it for? What are they doing with it? So with that, I thank you so much for your attention tonight, and I'm happy to take questions afterward. Apparently that's how it works here. So thank you.
That was a great talk. Oops, I got to turn this back on. That was a superb talk. It really makes me proud to be part of UMass, where so much cutting-edge research is done. Everyone is free to go, but we ask those of you who would like to ask questions to come to the front. If there are a bunch of you, then I'd advise you sit in the front rows, and Professor Patek will take questions from you as a group. If there aren't a lot of you, then you can speak to Professor Patek individually. I just want to present Professor Patek with a small group gift from the Commonwealth Honors College. Thank you so much for your wonderful talk. It was really perfect for the course. I want to thank Brian Dulick, and Alex Phillips for helping to organize the talk. And finally, thank you all for being such great students and coming out tonight. Enjoy the rest of the evening. I hope some of you will come up and continue the discussion. Thanks again.